Here's what shows up on your exam. One missing transporter protein can leave a virus-infected cell completely invisible to your killer T cells. In this video, we'll cover lymphocyte trafficking, MHC Class I and II pathways, cross-presentation, proteasome inhibitors, and antigen-presenting cells. And I'll flag the timestamps in the description below if you want to jump straight to one. But before any of that antigen ever reaches a T cell, it has to travel. So let's start with how antigen-presenting cells actually find their way to a lymph node in the first place. It all starts in the peripheral tissues, where dendritic cells pick up a pathogen. Toll-like receptor engagement activates the DC, and that activation switches on a chemokine receptor called CCR7. CCR7 binds two chemokines, CCL19 and CCL21, made by the lymphatic endothelium, and that gradient is what physically pulls the dendritic cell into the afferent lymphatics and toward the node. Naive T cells use this exact same receptor system. They also express CCR7 plus L-selectin, which lets them cross high endothelial venules and enter the node's paracortex, which is exactly where the antigen is now arriving. Meanwhile, if the antigen is blood-borne instead of tissue-borne, the spleen does the filtering. Antigens and APCs meet T cells in the periarteriolar lymphoid sheaths, or PALs, after passing through the marginal zone. And here's the clinical catch. If CCR7 or its ligands are defective, DCs can't migrate and T cells can't get primed, leading to immunodeficiency and a much higher risk of mucosal infections. Now that we've covered how cells get to the node, let's look at what they find once they arrive. The lymph node has three main zones. The outer cortex holds the B-cell follicles, both primary follicles and, once activated, germinal centers. Move deeper and you hit the paracortex, which is the T-cell zone. This is where you'll find most of the dendritic cells and the high endothelial venules we just talked about. In the center is the medulla, made up of medullary cords and sinuses, packed with macrophages and plasma cells. As for the plumbing, lymph comes in through the afferent vessels at the subcapsular sinus and leaves through the single efferent vessel at the hilum, a layout worth memorizing cold. With the lymph node mapped out, let's move to the spleen, since it handles antigen a little differently. Its white pulp contains the periarteriolar lymphoid sheaths, or PALs, where T cells sit, plus follicles for B cells, both wrapped around a central arteriole. The red pulp is full of sinusoids and macrophages, and its job is basically cleanup, clearing old red blood cells and any blood-borne pathogens. In between the two is the marginal zone, home to specialized APCs and marginal zone B cells, which is exactly where blood-borne antigen gets captured first. So putting the last two scenes together, antigen picked up in the tissue triggers DC uptake and CCR7 upregulation. The DC travels through the afferent lymph, arrives in the node paracortex, and that's the moment T cell priming happens. That's the complete trafficking route, start to finish, and it's the foundation for everything we're about to cover on antigen processing. Now that we know how antigen gets to the node, let's look at what actually happens to it once it's inside the cell, starting with the exogenous pathway. This pathway handles extracellular threats, bacteria, parasites, toxins, which get endocytosed and broken down inside lysosomes, and the resulting peptides are ultimately presented to CD4 positive T cells. Here's where it gets specific. A molecule called the invariant chain, or II, associates with newly made MHC-II in the ER and physically blocks the peptide groove, which is what routes the whole complex into the endosomal system. Once II is cleaved, a small fragment called clip is left sitting in that groove, which is why HLA-DM has to step in next, catalyzing the exchange of clip for a proper antigen-derived peptide. And this step is essential. Without a peptide, MHC-II is unstable and gets degraded, which is why a defect in HLA-DM causes bare lymphocyte syndrome, type 2. So that's how the outside world gets flagged to CD4 cells. But what about threats already living inside the cell? That's the job of the endogenous pathway. Here, intracellular threats like viruses or tumor antigens get tagged by ubiquitination, which marks them for destruction by the proteasome. The resulting peptide fragments are then carried into the ER by a dedicated transporter, TAP, the transporter associated with antigen processing. Once inside the ER, a molecule called tapasin bridges TAP to an empty MHC-I molecule, which is what actually facilitates peptide loading. Once that stable peptide MHC-I complex forms, it transits through the Golgi to the cell surface, where it's presented to CD8, positive T cells. And remember the pearl from the start of this video. TAP deficiency leads to low MHC-I expression, impaired CD8, positive responses, and a much higher susceptibility to viral infections. Quick check before we move on. Do you remember which MHC class presents to CD8, positive T cells, MHC-I or MHC-II? Drop your answer in the comments. We literally just covered it, so if you're confident, comment it now, and I'll circle back to it later in the pearls at the end. Back to the material, we've covered exogenous antigen going to MHC-II and endogenous antigen going to MHC-I. But there's a clever exception that bridges both worlds: cross-presentation. Here, a dendritic cell phagocytoses an entire infected cell or an apoptotic body, essentially swallowing an exogenous source of antigen. Instead of that antigen going down the usual MHC-II route, it escapes into the cytosol or gets routed to the ER through TAP-dependent mechanisms, which means it ends up loaded onto MHC-I instead. As a result, those MHC-I peptide complexes get expressed on the DC surface, priming naive CD8 positive T cells, and this whole process gets a boost from CD4 positive T cell help through IL-2 and CD40L. This matters because cross-presentation is crucial for antitumor immunity and for fighting off viruses that don't infect dendritic cells directly. So, where does that leave us clinically? It leads straight into a drug class that actually exploits this proteasome machinery, proteasome inhibitors. In rapidly dividing tumor cells, blocking the proteasome disrupts normal protein turnover, causing pro-apoptotic regulators like p53 and IB to pile up, and that accumulation pushes the cell toward apoptosis. The first-in-class drug here is bortezomib, approved for multiple myeloma and mantle cell lymphoma. Carfilzomib is the next one to know. It's an irreversible inhibitor used in relapsed or refractory myeloma. Put simply, the mechanism runs in a straight line. Proteasome inhibitor blocks turnover, p53 and cyclins rise, the cell cycle arrests, and apoptosis follows. In practice, watch for the side effects. Peripheral neuropathy, thrombocytopenia, and GI symptoms are the ones most worth remembering. Now that we've covered the drugs, let's step back to the cells running this whole system, the antigen-presenting cells themselves, and how they compare. Dendritic cells constitutively express B7 and CD40, along with constitutive MHC-II that's further boosted by interferon gamma, and their main job is priming naive CD4 positive T helper cells. Macrophages only express B7 and CD40 when induced, and their MHC-II is normally low or negative until interferon gamma turns it up, which is why their role centers on Th1 polarization and cell-mediated immunity. B cells constitutively express CD40, induce B7, and boost their MHC-II with IL-4, which fits their role driving Th2 responses and humoral immunity. Zooming into the co-stimulation itself, B7-1, also called CD80, and B7-2, or CD86, both bind CD28 on the T cell, and this interaction is essential for T cell activation. The other key pair is CD40 on the APC engaging CD40L on an already activated T cell, which enhances APC function and cytokine production. Building on everything we've covered, let's pull it all together into the pearls most likely to show up on your exam. MHC-I is expressed on all nucleated cells and presents endogenous peptides to CD8 positive T cells. MHC-II is restricted to professional APCs and presents exogenous peptides to CD4 positive T cells, and that's the answer to the quick check from earlier. CLIP is removed by HLA-DM, and if that step fails, antigen presentation is defective. TAP and tapasin are both essential for MHC-I peptide loading, and dendritic cells are the only APCs that can cross-present and prime CD8 positive T cells. And don't forget CCR7. It guides both dendritic cells and naive T cells into lymphoid tissue, and any disruption impairs immune surveillance. One more self-check. Peptides from cytosolic viruses are transported into the ER for MHC-I loading by which complex? The answer is the TAP complex, and if you got that, you've got the whole endogenous pathway down. So that's the complete journey, from antigen picked up in the tissue, through processing on MHC-I or MHC-II, all the way to T-cell priming in the node. If this helped, like the video, subscribe for more high-yield immunology, and don't forget to drop your knowledge check answer in the comments below. Thanks for watching.